I'm going to talk about clinical trials in the era of precision medicine a little bit more generally than the last talk, and I'll use glioblastoma by the end of the talk as a specific example for some clinical trial design choices that we made. But hopefully by that point, you can see why we made those choices and why it may differ a little bit from the things that you've heard before, so that you can understand, I guess, when new ideas come up or new design elements come up, what are the considerations that you have to take into account when you're making these choices. So we've heard a couple of precision medicine definitions so far. Again, I'll be more general. I think that this era is about tailoring medical treatments to the individual characteristics of each patient. And these are, to do this, we identify populations with different susceptibility to disease, biology, prognosis, but mostly response to a specific treatment. And I think a lot of the times we'll talk about this with respect to targeted agents because that makes the most sense biologically and with a priori biologic hypothesis. But really any biomarker that's predicting a response or an outcome is something that we could consider precision medicine, even if it's not a molecular one. And so for clinical trial development, what this means is that we need to develop the biomarkers and information about the biomarkers in parallel as developing information on the therapeutic, and it also increases the testable therapeutic hypotheses. So this is a slide I use for glioblastoma, and on the right is a review from New England Journal, now 10 years ago, looking at targeted agents with different targets for glioblastoma, and on the left is the TCGA paper from Nature in 2008 outlining genomic alterations in glioblastoma. And in the last talk we heard, this isn't like lung cancer. Well, glioblastoma really isn't like lung cancer. You can't have a pie chart that kind of segments the population and there's one biomarker that has a driver for each one. There's really overlapping biomarkers, and we really have no idea a lot of times how those pathway interactions and aberrations interact with the therapies. So a lot of a priori hypotheses in terms of driver mutations and blocking the kind of downstream impacts of driver mutations are valid, but to kind of extend that to every kind of targeted agent or every kind of situation gets you into trouble a bit, and it certainly does in glioblastoma. So this is from a review, and this is basically a review of different ways generally that you can think about how to set up a biomarker study. So A is pretty common, thought of in basket trials. You test the biomarker, and if you're positive, you get enrolled, and then you put everybody on the experimental therapy. In B, you test the biomarker. If it's positive, you randomize to experimental arm versus control. C, you test the biomarker, and while you have the biomarker information, you still randomize everybody to experimental versus control. And then there's some other kind of ways you can do it based on biomarker strategies, but these three are the ones that I think I'll focus on the most, and the choices that go into which one is the best one for you. And certainly putting these components into platform trials is the next step. So why do people care about biomarkers outside of the biology? This is a thought experiment that was published in JCO, and basically what it said was, imagine a situation where there's 10% of the population that will survive 5 times as long if they get a drug, but you don't know who those people are. So if you just run this unselected population in a trial, you would have 334 patients on each arm, and you'd have by traditional measures an insignificant p-value, even though you've enrolled all those patients. If you could select those patients, though, you'd only need 16 total patients and you'd get a positive study. So this is kind of like the big signal in a small biomarker frequency population, which is the dream of biomarker-directed therapy or precision medicine. Short of this kind of extraordinary situation, the FDA has published guidance on the use of biomarkers in clinical trials. And this is looks like a busy chart, but it's really helpful. Basically, this column says what frequency of the overall population is your biomarker present. So is it all of the people? Is it 25% of the people? In the example that I just showed, it was 10% of the people, so it's way down here. And then these two columns say, what is the effect size in the biomarker negative population? So is it a predictive marker, but there's still effect in the biomarker negative population, it's just not as great? Or is it a completely predictive marker where if you don't have the marker, you don't respond at all? And what the values here are what they're calling sample size ratio, or how many more times patients you need to enroll to have the same power of the study. So not surprisingly, if everybody has the biomarker, it really doesn't matter, it's the same amount. And in the best case scenario on this graph, 25% of the patients with no effect in the biomarker negative people, you get 16-fold increase in efficiency by finding those patients. But here, like if the biomarker frequency is higher and there's still effect in the biomarker negative population, you get much more modest gains in efficiency. So I think this is really important when you're thinking about whether you want to do a biomarker selected trial, but it's also important to think about like the incentives for people to develop biomarkers. So glioblastoma, we talk about it being a rare disease, and certainly some fusions within glioblastoma are very rare, even within glioblastoma. But finding small biomarker populations is the name of the game for drug development for biomarker companies and for companies like Loxo that want to develop biomarkers for small populations. It's not worth finding a biomarker if it's 75% of the population. It really is critical to find the biomarker if it's a very small frequency of the population. So this is an example of how this would work, how the kind of biomarker selected design would work in real life. You'd do some kind of biomarker assessment. You'd enroll everybody on the experimental therapy and look at the outcome. But then all these patients that are biomarker negative are off study. And I think we saw a great example just now of like how you can get efficiencies by having a platform trial with some common screening procedures. Even the last two talks is that you don't want to do screening for 5% of the population and then 95% have nothing to go on, and you've screened all those people and then they don't have any clinical trial options. So you could do therapy B, therapy C, therapy D, and do all these kind of things kind of in parallel with each other. This works really well in the situation where the outcome has very little variability, and we'll get into that a little bit, and the biomarkers are kind of mutually exclusive and exhaustive of the entire patient space. So if you have one biomarker, you don't have another, and you can take every patient and kind of divide that up, which was kind of what the hope for lung cancer is, this works great. It works even better if these biomarkers have no prognostic value at all versus each other. Because if these biomarkers have no prognostic value, then this patient that comes in, you don't know their biomarker profile, there's like limited selection bias on what arm they go on. So you could argue that you could compare these versus each other in some way. Some statisticians in the room may tell me this is a bad idea. But if randomization's purpose is one of the purposes is to limit selection bias, having a patient that you don't know their biomarker profile and having an algorithm to assign them to a specific arm, how are you selecting patients? I mean, it's not done in advance. So we heard about platform trials, and some of the benefits from platform trials. I just kind of restated this kind of multiplex biomarker testing efficiency. We heard about dropping and adding arms without reinitiating the trial, and this is huge. I mean, I echo your comments. I was frustrated when we're trying to do trials for glioblastoma when I first came on staff because we were asking the same questions every time, and it would take 6 months to get everyone together to really talk about it and like have the meeting and the call, and not everyone can get on the call until Thursday next month and all these kind of things. But we really just cared if the drug was improving overall survival for glioblastoma. So why do we recreate the wheel? The other thing for us was that, and I'll get into a little bit about what kind of endpoint you're selecting, but we really wanted control arms for our endpoint, and so conserving these control and having one control arm for multiple experimental therapies makes patients happier than one-to-one randomization and also provides a lot of efficiency. And then finally, which I won't talk about too much, except for outcome adaptive randomization at the bottom, is there's opportunities for other innovations here. And Don will talk more extensively about these, but financing structures about how to keep these going, that's kind of a need and not just an opportunity, learning systems, and then outcome adaptive randomization. So outcome assessment. So this number doesn't really mean anything. So I was taking for glioblastoma, I was taking it as a median survival, but it could be anything. basically is just a value of an endpoint. And what's saying is we test the biomarker and we get an experiment and we put them all on an experimental therapy, we'll get some result. We'll know that the patients that got treated and have the biomarker lived a certain amount of time. You could use, response rate or whatever you want here, but you don't know what people that weren't treated would have got or biomarker negative. So this is going to be like real 101 stuff, and it may bore you, but I think it's important. If the patients would have had the same result, whether they were on control or got treated, as long as they had the biomarker, that's a prognostic biomarker. So you don't know whether your biomarker is having this effect or whether your drug's having this effect. If it doesn't matter whether you have biomarker positive or negative and you just needed to get the treatment, you would have this situation. And if it's really a predictive biomarker, you only get the benefit from the treatment if you have the biomarker. And so if you just have this, you can't fill in these boxes and you don't know which one you're in. Now you may have really good historical control data and you may have no variability in your endpoint so you can estimate it, so you could fill these boxes in with things outside of the trial. But for any endpoints with kind of variation, randomization becomes more and more important. So we thought, well, this is to signify randomization. That's thermal noise, so it's a random process. So you could take everybody with a biomarker and randomize them to experimental versus control and look at their outcome. This is kind of like lung map, having control arms but still having this biomarker assignment. When you do that, you can kind of knock prognostic off the ledger, and you still have treatment works versus a predictive marker. But the other issue is, and again, I've said this before, but this is our TCGA data and our data. We were looking at different pathway alterations because we were designing a platform trial for glioblastoma for these specific biomarker categories, and we thought, okay, do they overlap? So we have an EGFR biomarker, a PI3 kinase biomarker, a p53 biomarker, and a CDK biomarker, and each of these biomarkers have rules. But when we looked at the overlap of these in TCGA data and our data, there was a substantial amount of overlap. So if you had one, they weren't associated with, or they weren't correlated with each other at all, really. If you had one, you could have two, you could have three, you could have none. And how do you assign patients to each one of those? Furthermore, as we said before, we don't know whether the biomarker with a targeted drug to that biomarker is better than being on an immunotherapy, is better than getting radiation, is better than getting standard chemotherapy. We're making a value judgment of what we think the best treatment is before when we create any kind of algorithm, and that's what the clinical trial data was supposed to show us. So what happens if we move randomization up front and randomize to control versus all the experimental agents and compare these all versus each other? We would get the best information about the biomarker, but this may be the most inefficient way to do it. So, people, when we started describing this, even in glioblastoma, when we started describing this design, people would say, well, if they have EGFR amplification, why aren't you going to put them on an EGFR inhibitor? Like, that's crazy. Like, it would be immoral not to put them on an EGFR inhibitor. And I said, where's the evidence that that works at all in glioblastoma, or that it works better than anything else? So you get a lot of pushback from this, even in like glioblastoma, where you have no real evidence that the biomarker selected treatment is the best option. So I said I gave you the best information, but it's really inefficient. So what can we do to kind of bridge that gap a little bit? Well, one of the things we can do is do something called an adaptive trial, and there's lots of different kinds of adaptations that happen in trials, but I'm going to talk mostly about adaptive randomization. I'm not going to stick on this too long because this is Don's thunder, but these are the first two publications from the I-SPY 2 trial in the New England Journal. So this is Bayesian adaptively randomized trials gone mainstream. And I think that the really interesting thing about this, other than all of it, is that if you look at the results, in the New England Journal, you're seeing a probability distribution. This isn't like a hypothesis test. It isn't like, the p-value. You see a control arm, and you see the neratinib arm, and the estimated response rate probability distribution. And we can make We can make judgments on how overlapping or non-overlapping we want these to be before we decide the trial's over. But what they calculated, and Don probably will go into in more detail, is the predictive probability of a canonical phase 3 study, which they estimated at 79%. And that's how the trial went. So if you have a probability distribution that's building up over time during the course of the trial, can you use that probability distribution to weight your randomization? Of course you can. And that's what Don did in I-SPY. So basically what we could say is we start the trial and we have this kind of situation where we randomize everybody no matter what biomarker they have, because we want to be agnostic and we don't want to have hubris when we're trying to understand biomarkers in the relationship to therapeutic effects. But during the course of the trial, if we see that the treatment effect is biomarker specific, it kind of evolves over the course of time to a biomarker specific trial. And so this is how we apply this in glioblastoma at Dana-Farber, an investigator-initiated study. This is called INSIGHT. We took newly diagnosed glioblastoma, did profiling. We randomized everybody to a control arm or 3 different experimental arms. And then we used the accumulating results of the trial to inform randomization, to weight randomization if one of the arms is doing better than other arms. I'm going to have a movie, I think. OK, this is the time to click on the movie. There it is. OK, so in the left, we have a Kaplan-Meier curve. This is the data from the trial that's accumulating. And that data is being used to estimate. It's not really in great order. That over here is being used to estimate a posterior probability of a treatment effect for each arm. Those treatment effect estimates then go and weight the randomization. So right here, the best example is this red arm that's doing the best. We have a pretty high probability that that's working. And every patient that comes through, this is their biomarker profile, positive, minus positive. These are, I mean, the trial's over now in this simulation, but these are the randomization probabilities. So the control that we held constant to the best performing arm, the red arm, because that one's the best, and then the green and the blue have pretty much dropped out from accruing any more patients because they're not doing so well. So that was INSIGHT is up and running as a phase 2 screening trial with Dana-Farber as the main site and 9 other sites. Then working with Don, we put this on steroids a bit and said, well, what happened if we don't just do newly diagnosed, but we do newly diagnosed and recurrent glioblastoma, and we consider that indication a biomarker? And I told this before, like when Don said, oh yeah, it's a biomarker, newly diagnosed for recurrent, I was like, that's crazy, that's not a biomarker, that's a biomarker is like P53. And he said, no, it's the same thing if you put it in the model. And what it really turns out is 2 things. One is that if a drug is working equally well in newly diagnosed and recurrent, then you have a doubly powerful trial just by combining those trials into one trial. And 2, you can create a situation where if this trial is open and the platform keeps going, then patients can come into a clinic and just enroll on GBM-Agil. Whether they're newly diagnosed or recurrent, we're just learning as much information as we can from them, every patient that comes in. And this is a busy schematic, but basically it just shows the process of how patients get enrolled, and we use their information to update the estimations of the treatment effect, and we use those estimations to make decisions about whether to keep the arm going or not going. The other reason this gets put on steroids is because we went and we talked to the FDA about this and to the previous point about registration of drugs, and we said, well, we're using overall survival as an endpoint. If we have a signal and it looks like there's a drug that's improving survival in glioblastoma, would you be comfortable approving the drug based on this Bayesian adaptively randomized stuff? And they said, I think we're getting there, but what if we compromised and had another stage where you had a fixed randomized set, just so we can have a better idea of type 1 error. And so we agreed, and Don designed a trial that had this Bayesian adaptively randomized screening part, and then anything that looks like it's working really well goes into a limited confirmatory cohort, not a big phase 3 trial, but using all the data from the first adaptively randomized part and the additional data, and they said that would be, we could use that to go and submit for registration and marketing purposes, and they gave us a letter to codify that because none of the companies believed us when we told them. Or they wanted to be really sure, let me just say it that way. So finally, one kind of thing that we're kind of running into a little bit is that a lot of times we have the opposite problem, and people think, well, I have this EGFR V3 antibody, like why would you put anybody on this drug if they don't have EGFR V3? And so I said, okay, well, what can we do this kind of thing in the reverse? Start enrolling just a biomarker positive patients. But what if that cured all the GBM patients? I mean, if you were wouldn't you rather try that? Maybe your assay was wrong on V3. Maybe the hypothesis was wrong. I mean, if the signal was so big, let's not just assume that it's only limited to that. I mean, compared with the rest of the thing. So, thought about doing this design called a Bayesian basket, where you can have a traditional basket study that stays biomarker specific, a balanced randomized study that is randomized the whole time. There's adaptive two-stage design where you can kind of make an interim assessment whether you want to stay biomarker specific or not. We thought, well, what if we're doing it in a platform trial, why don't we make those decisions arm specific based on the level of evidence of a biomarker specific therapeutic effect? So if you do have some things that are very biomarker specific, and for Insight, there's a drug, AMG 232, which is an MDM inhibitor, in phase 1 trials that we might want to put in there, and that's a very p53 mutant. There's no effect, and people would it be ethical to put those patients on? Well, maybe that arm stays biomarker selected the whole time. But all these other ones that are kind of complex, maybe start more biomarker selected, and then if we're wrong, it becomes randomize everybody, or we start randomize all and it becomes more biomarker selected. So I think I'll end there, and hopefully a good segue into what Don will talk about, and we'll go from there. So thank you.